Hey guys, I just wanted to go over with you real quick the notes over the cell cycle. And so you can watch this video either today or sometime the next couple days to take some notes over this. So you'll have those in your notes. And tomorrow in class, we'll probably get started on another assignment over the cell cycle. So I'll try to get this covered as quickly as possible. So when we talk about the cell cycle, just the regular pattern of growth, DNA replication, cell division that occurs in the eukaryotic cells. We can just think about it as the life of a cell. Okay, that's what the cell cycle is. So from the moment that it begins to where it will split into two new cells, and that is the cell cycle. And we're going to go through all the parts of it. So the growth and the DNA replication are going to occur during what we call interphase of the cell cycle. And that's going to be most of the cell cycle's life is going to be in interphase. 90% of the time, the cell will be in interphase. And that's whenever the cell is doing its job, it's doing, we'll talk about it in more detail, but when you look at this, this is a microscopic image of probably an onion root cell, and hopefully we'll get a chance to get the microscopes out and look at these this week. But you'll notice a couple things I want to point out about a cell that's in interphase. You have this very distinct cell membrane separating the contents of the nucleus. I said cell membrane, or nuclear envelope, separating the contents of the nucleus to the cytoplasm. And then within that nucleus, you'll see 1 to 2 nucleoli, nucleolus or nucleoli if there's more than one. And that's those darker spots in there. So when we come and look at this, this is kind of a graphic representation of the cell cycle. You'll notice that most of the cell's life is in interphase and all the way here. And then you get what's called the M phase or the mitotic phase. And that's where mitosis, which is nuclear division, and then cytokinesis, which is the division of the cytoplasm, occur in the mitotic phase. So most of it is in interphase, and we'll go through real quickly and look at these three subphases of interphase. The first one is G1. This is where cell growth is going to occur. Whenever all the organelles could be replicating and growing inside the cell, and then this is basically when the cell is doing its job, whatever that cell. Maybe it's making a certain protein, and it's going to be doing that job throughout. Typically throughout the G1. Then we come to the S phase of interphase, and that stands for DNA synthesis. So in DNA synthesis, what's going to take place is all of those chromosomes are going to be replicated. So remember, you have 46 chromosomes in your nucleus in our cells, and each one of those 46 will then be replicated. And when one of those chromosomes replicates, it replicates into two identical what we call chromatids, sister chromatids, and we'll talk more about that briefly here in just a moment. But this is an important phase because if we have 46 chromosomes in a cell and they were to split into two cells, we would have 23 in each of the new cells, so that's not going to work. We need the two daughter cells to have 46, so that is where this replication comes in. It's very important that each one of these chromosomes makes a copy of itself. So for a short time there, you actually would have 92 chromatids, and then those chromatids are separate into 46 and 46 in the two new cells. Hopefully that makes sense to you. Then the last phase of interphase is just simply G2, and that is when the cell is getting ready for division. Some checkpoints are basically examined and make sure that everything's ready and all the chromosomes have replicated and everything's good to go for cell division. So that leads us into, well, before we get to mitosis, let's talk about this real quick. So cell division of mitosis is going to result in genetically identical daughter cells. So all of the cells in your DNA, which is called your genome, DNA molecules are packaged into chromosomes, and somatic cells contain a full complement of chromosomes. So mitosis is going to occur in all of those somatic cells in your body, which is every cell in your body except your gametes here, which are your sex cells. And that is, by the way, we'll talk about it more later, but that is where meiosis takes place. So somatic cells, all of the cells in your body are going to undergo mitosis. Gametes will undergo meiosis, and we'll talk about meiosis. Probably won't be until January. But the key thing here is that cell division is going to result in two daughter cells that are genetically identical to each other and identical to the original parent cell. So first of all, we're going to talk about what is mitosis. Mitosis is simply the division of the cell's nucleus and its content. Okay, that's what mitosis is. And there are four phases that we're going to be talking about and going into a little more detail. The first phase is called prophase. Chromatin is going to condense into chromosomes. You start to see those chromosomes for the first time. They're referred to as sister chromatids at this point. Centrosomes move away from each other as the spindle starts to form between them and the nuclear envelope starts to break down. So if we look at a couple of these images here, this shows you, so this one chromosome has replicated into two sister chromatids. So these two chromosomes or chromatids here are identical to each other. These two are identical to each other and so forth. And you'll notice the membrane starts to break down the nuclear envelope. You can look at this under a microscope and you can't really see the actual chromatids, but you can see that chromosomes are starting to condense down, and you can see those darker. You don't have that solid circular nucleus anymore. You start to see individual chromosomes, and you'll notice that the membrane here is starting to break up. That is a key feature of prophase. Second, another key feature of this is that mitotic spindle. It's going to consist of microtubules, and remember, microtubules are part of the cytoskeleton. We learned about that a little bit when we talked about cells. And they play a major role in mitosis. Their job is going to be pulling those chromosomes apart from each other. So you can see the spindle, the mitotic spindle is this green here, and these are your chromosomes, and they will be pulling them apart. And there you can see them in the microscope, the spindle there, pulling those chromosomes apart. Don't worry about that. And we're going to not worry about prometaphase either. We're going to jump straight to metaphase next. This is considered usually the longest stage of mitosis, and this is a real simple one, easy one to recognize. Your chromatids are lining up in the middle of the cell or at that metaphase plate. If we were to look at them, you can see them lining up end to end with each other there. And it kind of looks like this under a microscope. I've always kind of think it looks like, if you've ever gotten stitches before, it's kind of what it reminds me of. But you can see they kind of line up, and you'll notice that the nuclear membrane is completely gone. You can't see it any longer. So that is a key feature. And a lot of times when we look in a microscope and if we're looking at the different phases of mitosis, you'll see more metaphases, probably easier to find than the other phases. Sometimes prophases are pretty easy to find too, pretty numerous. The third phase is anaphase. So we've had prophase, metaphase, and then anaphase. This is typically considered the shortest phase. Sister chromatids are going to now, at this point, they are now considered chromosomes again because they get separated. So the easy way to remember anaphase, I think of, is the chromosomes are pulled apart. Anaphase apart. And the cell will typically begin to elongate a little bit, and you can see that here in these images. And notice how these chromosomes are getting pulled apart from each other. And now they're no longer sister chromatids; they are now officially chromosomes. The last phase is telophase or telophase. I've heard it pronounced both ways. Your two daughter nuclei begin to form. Nuclear envelope reappears and the chromosomes decondense. So hopefully you'll notice that sounds very similar to prophase. It's just the exact opposite. So in prophase, the chromosomes condensed, the chromatin condensed to chromosomes, nuclear envelope started to break up, and here just the opposite is taking place. Nuclear envelope is going to reform and the chromosomes decondense, and it will start to that nucleus will start to get really dark. Now in plants, you'll notice that a very distinct feature is that the structure called the cell plate will begin to form. We'll talk about that here in just a second. So this is a plant cell under that. This is that cell plate. It's the precursor to your cell wall, and you'll start to see that. Notice how dark the nucleus is starting to get. That's because all those chromosomes are decondensing, and you're starting to get more of a circular shape to that nucleus. Key feature to telophase. Cytokinesis is after mitosis, is the division of the cytoplasm. It's a little different between animals and plants. In animals, what's called a cleavage furrow will form, and that's just kind of where the cell membrane will kind of pinch in, and eventually it will go meet all the way, and when it contracts all the way in, it will separate those into two new cells. In a plant cell, you start getting this cell plate beginning to form, and you'll notice in this picture that cell plate, once it reaches one end of the cell to the other end of the cell, it's no longer a cell plate, it's the new cell wall, and you have two new daughter cells. So, but as long as that cell plate is not completely across there, you still just have that one cell with that is in telophase. And so there that. The last thing, well, one of the last things real quick I wanted to go over with you. This is just some images so you can recognize the different phases. So if we notice interphase, very distinct circular shape of the nucleus, and in some of these you can see that darker nucleoli in there. Prophase, you'll notice that this chromosomes start to appear. Here's a good prophase, here's a good one. Notice how the nuclear envelope starting to break up. Metaphase, again, you're looking for where those chromosomes line up in the middle. Here's a good metaphase. Here's another metaphase. Here's one here. And then anaphase, they start to pull apart. There's a good anaphase there. There's a very early anaphase, another anaphase right there. And then the last phase is telophase. And what you look for here is, this right here is probably really late telophase. This is a good telophase right here. See how dark the nucleus is getting. And you probably can faintly maybe see the cell plate forming between there, maybe right here too. Sometimes it's tricky to tell the difference between telophase and whenever you get two new cells because there's a thin line between the two. The last thing I wanted to go over with you real quick was, and you had the hyperdoc on it last week of cancer. So it's real briefly I want to talk about cancer. Is a disease caused when cells divide uncontrollably and they spread into surrounding tissue. So typically what happens is the cell will go from mitosis to mitosis to mitosis to mitosis. It never goes into interphase where it will typically do its job. So you have these cells just dividing uncontrollably, and they are not able to do their typical job. A couple features of cancer that I want to talk about is this subject called density-dependent inhibition. It's a process in which crowded cells will stop dividing. So if cells are starting to get crowded together, there's no room for them to go, so they won't divide. And then there's this other feature of cell division called anchorage dependence. Anchorage dependence is whenever to divide, most cells, let me go back to that. Most cells must be attached to a substrate. They have to be attached to something before they can divide. Cancer cells exhibit neither density dependence inhibition nor anchorage dependence. So if you look at this picture here, these are cells that were grown in culture in a little petri dish or this little flask. And you'll notice typically a cell will reproduce as long as there's room. So they will fill up the bottom of this. And that's where that density-dependent inhibition is going to kick in. If you were to take some of those cells out, they would fill in, those in the middle would fill in that space. Now, the other thing, notice how they're just one layer thick. But when you get to cancer cells here, I'm going to zoom in a little bit. Cancer cells usually continue to divide well beyond a single layer, so you're going to get multiple layers in there and forming a clump of overlapping cells. They do not exhibit anchorage dependence or density dependent inhibition. So what that means, they could get in cancer cells don't have to be attached to a substrate to reproduce. They can be in your bloodstream. Cancer of your white blood cells, leukemia, are probably going to be doing things similar to that. So that's a little bit about cancer, and hopefully you've understood a little bit about that in your hyperdoc that you did last week. So I'm about out of time, so I'll see you guys in class tomorrow. Hopefully you get a chance to watch this today, and feel free, and if you have any questions, have them available, and we will discuss this in class tomorrow. See you later.